All right, A&P class. Well, we're going to dive right into this next set of notes, which is the immune system overview, as well as innate immunity. Okay, so let's get going. I will send you this link so you're able to watch this immune system overview video, which is just kind of a nice intro to the whole immune system. Okay, so let's start out with the levels of immunity. Okay, again, we have what's known as our innate immune system and our adaptive immune system. Okay, and so this chart from an old version of the book is a nice view of this and kind of nice depiction of this. Okay, so the innate is non-specific; it just attacks everything in the same way, whereas the adaptive is specific, specifically destroys invaders. Okay, so with our innate defenses, the first line of defense are our surface barriers. Right, this is just stopping things from getting into the body. Second line of defense is our cells and other things that attack things that get in. So phagocytes, natural killer cells, our whole inflammatory system, antimicrobial proteins, and then the process of fever. And we'll talk about all of this in more detail today. And then the adaptive system, we have two levels of that. Humoral immunity, which is really driven by the B cells and our antibodies. And then our cellular immunity, which is driven by our T cells. And both of these are called lymphocytes because they're mostly found in the lymph, B cells and T cells. And we'll talk about these more tomorrow, or next class. Next class. Okay. So a couple things on innate versus adaptive. Again, innate is non-specific, adaptive is specific. Innate attacks all foreign invaders. Okay, it has no memory. This is where fever and inflammation come from. It uses phagocytosis, natural killer cells, and antimicrobial proteins. Okay, the adaptive system usually needs multiple exposures to build up defenses, uses cytotoxic T cells, and antibodies. Those are the two primary weapons. Okay, it uses B cells to build up immunity and to make antibodies. B cells are the ones that make the antibodies. Okay, and again, they're they're not completely separate from each other, and they do work together. And you'll see some ways where that happens. Okay, so let's dive into this idea of what an antigen is. An antigen, the definition, is a substance that can mobilize the adaptive defenses and provoke an immune response. The root of the word is antibody generating substance. Okay, so antigen, right? Something that causes your immune system to generate antibodies. All right? And there's different types of antigens. First of all, there's self-antigens. And self-antigens are important because they allow the immune system to see if something is supposed to be in the body or not. Okay? And the primary ones are called major histocompatibility complex. So what's the root of this? Histo, just like histology, right? So this means tissue. And then compatibility, right? Is that tissue compatible with the body? Is it supposed to be there? Okay. Function of these is this is how the immune system distinguishes self from non-self. Okay. And so we have two major types of these MHC proteins that stick out of all cells in the body, or most cells. Okay, the first one is called MHC1. These are proteins expressed on all cells except for red blood cells. And think of this as the passport. Okay, so when you think of this as kind of a travel analogy, when you're in, you have to have some proof, some document when you're in the country here in the United States. Most of us all have some proof of the fact that we're a legal citizen of the United States. Okay, and so this might be our passport, our birth certificate, something like that. Okay, so think of this as a way for the body to tell if something is legally supposed to be there or not. Okay, MHC2 proteins are expressed only on immune cells, And think of this as a law enforcement ID. Okay, so these proteins are only on immune cells. Think of this as like the sheriff's badge or something like that. Okay, and so this depends on genetic inheritance, and there's millions of combinations possible. Okay, interestingly, identical twins have the exact same MHC proteins, and there's some similarities among relatives. Okay, so just basic definitions here on antibodies. Antibodies are proteins, also known as immunoglobulins. They're part of our plasma proteins that are just always in the blood, right? They're always floating around. Okay, so protein made by the B cells specifically in response to an antigen. And so what they do is they bind to that antigen and either inactivate it or help to destroy it. They could destroy the cell that it's a part of, or they could destroy the antigen itself. Okay, the structure of these are there's usually some type of Y shape, looking somewhat like this, with the stem here, and then two binding sites. Okay, and so those are specifically a part of the adaptive system. Okay, only they're not a part of the innate system. And the interesting part about this is, so again, these are made by B cells, and they're made specifically designed to just destroy one type of antigen or stick to one type of antigen. And so by the time we're adults, we're into our late teens, early 20s, and our immune system is fully developed, the body, each of us can produce about 1 trillion different types of these antigens, right? So we have memory B cells that are there specifically designed to make one type of antibody that's specifically for one virus, right, or one bacteria, or one other type of invader. And we have about around 1 trillion different types of those by the time our immune system's fully developed. Crazy level of complexity and just a vastness to the immune system. It's the least understood system in our body, second only to the brain. Okay, so T lymphocytes, which are T cells, so we're going to go through some of the cells related to all the immune system here. Okay, so this T cells are part of the adaptive system. They drive the cell-mediated response, not the humoral response. Okay, so they're produced in our bone marrow, but they mature in the thymus, and this is where they get the name T cell. Okay, they're sent to the bloodstream or lymphatic tissue after they mature. Okay, so remember that thymus was that big tissue, a big gland around the trachea that pretty much goes away in adulthood. Okay, the function is to recognize an antigen. T cells will recognize an antigen in combination with MHC protein, and they activate the immune response. So we need these to activate the T cells and the B cells. Okay, and so that's mostly driven by the helper T cell, that whole activation part. Okay, so we have multiple T cell types. Helper T cells, first to be activated, activates others. Cytotoxic T cells or killer T cells. These are the ones doing most of the work on the T cell side. These are the ones actually destroying things. We have regulatory T cells, which limit the response, slow things down, and then memory T cells, which are going to be there, kind of remembering what that antigen looks like, remembering how to destroy it if it comes in again next time. And then we got B cells. B cells are a part of the adaptive system as well. They drive the humoral response. They are made in the bone marrow, but then they also mature in the bone marrow, and that's why they're called B cells. Okay, so their main function is to produce antibodies, just like this. Okay, each only produces one type of antibody, right? So this is where we have that 1 trillion number of different B cells, each one making a specific type of antibody. Okay, plasma cells are really what a B cell is called when it gets activated. Okay, so we got B cells normally in our lymphatic system, right? That's why they're lymphocytes. Okay, but then let's say there's a B cell that's there for a certain type of flu virus. Okay, a specific type of flu virus. It's sitting there. All of a sudden, that virus that exact flu virus is encountered into your blood. All right, so then we have some cells that are going to go in and wake up those specific B cells, activate them. They're going to make their way into our bloodstream and start releasing antibodies. Okay, so when they're in this state, they're known as a plasma cell. All right. Okay, so a little leukocyte recap here. When we're looking at these different cell types, this would be something for you to fill out. We can just run through it here. This is has the functions and the which system they're a part of. So neutrophils, and we talked about a lot of these already back when we talked about the components of the blood. So I'd like for you to go in and look at those notes as well. And obviously you can pause this here and fill this in, but make sure you know the general functions of each of these cells and which system they're a part of. Okay, so let's dive in a little more specifically to the innate system. And so again, the first line of defense is just blocking things from getting in. And we've already talked about the skin and mucous membranes quite a bit. So we've got those keratinized cells of the skin, we've got oils on the skin, and we got mucous membranes. All of those things are helping to stop invaders from getting in. Okay, what about chemical defenses in the body? Okay, so how does a virus infect? Well, this is very relevant right now, talking about the coronavirus and what it does, but it's similar to other viruses in the way that it works, where it doesn't actually have a cell itself. It doesn't have the ability to make its own, to make proteins and replicate itself. So it has to go into the host cell, and it hijacks its metabolic machine. It hijacks its protein formation system, and it causes that cell to reproduce, or causes that virus to reproduce itself, and then just starts sending out all these other viruses all through the body. All those go in and infect all the other cells in the body, and then those start reproducing themselves, and it really taxes the body. Okay, so but when a cell is infected, one of the defenses that it puts up is it releases something called interferons. And interferons are a chemical that goes to the neighboring cells and causes those cells to put up a wall of defense, put up these virus-blocking proteins. They're going to kind of protect it. It's kind of like an alarm, like, hey, there's a invader on the loose. Lock your doors, close your windows. Right? That's pretty much what's happening. We also have what's known as complement proteins. These again are normally circulating proteins through the blood, so part of our plasma proteins. But they can become activated, and when they do become activated, they bind together and form pores like this in an infected cell, causing that cell to be destroyed. Okay, and they also activate the immune cells. So those are two just kind of chemical defenses, those interferons and those complement proteins that are always there. Okay, so the other cells, we've already talked about these in the blood, composition of the blood notes: neutrophils, natural killer cells, okay, mast cells, macrophages. Just a brief review of those definitions there. Okay, so let's talk about local inflammation and what that looks like. Okay, so there's really 4 classic signs of inflammation, and those are heat, redness, pain, and swelling. Okay, if you're a kinesiology major and moving into sports medicine in some way, you'll most likely run into a 5th one, which has to do with impairment of function. Okay, the 4 classic ones are here, but then that 5th one is some level of impaired function. Okay, so when we look at these signs of inflammation, what I'd like you all to understand is not just the definitions of the signs and what they are, but what's the physiology behind leading to each of those signs. Okay, so we're going to walk through that now through these next 2 slides, as well as a flowchart slide that puts it all together. Okay, so what starts the whole process of inflammation? It starts by damaged cells, right? So damaged cells, some cells get destroyed or messed up by a virus, by a bacteria, by an injury, right? By a splinter going into your skin, right? That could be one way of starting it. also could be mast cells interacting with something that activates them and causing histamine release. Okay, so histamine, that would be coming from the mast cells, but then damaged cells are going to release other chemicals, like just everything that's inside of them. Cells that are in, or chemicals are inside of those cells that are supposed to stay inside the cells and not come out. When they do come out, they activate the immune response. Okay, so what does histamine do? Histamine is a direct vasodilator. Okay, so that if we send more blood to an area, that's going to lead to that area getting warmer, as well as red. Okay, we also have other chemicals that stimulate pain or nociceptors. Okay, and some of those other chemicals are chemicals found inside of cells. There's one specifically called substance P that we find inside of cells. When it gets out of the cell and binds to pain receptors, it causes pain. Okay, so when we get that vasodilation, which is caused by histamine, what does that do? That delivers more white blood cells, delivers more neutrophils, monocytes, which are the ones that are going to turn into macrophages. Okay, so we deliver more leukocytes or white blood cells. We also deliver clotting substances so we can stop bleeding. So that's kind of the purpose of the vasodilation, right? As far as delivery goes, but then vasodilation is also important because of carrying stuff away, right? So we want to carry away toxic products and dead cells. So some of those things that stimulate the immune system, that's substance P, a lot of other chemicals that are inside of cells that are don't need to be there, get carried away. And then we also have all the just dead cells that were destroyed with the injury. That stuff needs to get carried away as well. Okay, so then other chemicals that were again released from cell contents lead to what's called increased capillary permeability, right? So that the ability of the capillaries to let things go through and get out of the bloodstream into the interstitial space increases. So this allows fluid, proteins, white blood cells to get into the interstitial space. This is good because we need that to clean out the space. And this will increase our lymphatic flow. Also delivers important things for clotting and healing, but this also leads to swelling, right? If we're adding more things to the interstitial space, that's how we get swelling. Okay. Also, some other things that happen when we deliver more immune cells there, we get increased phagocyte migration. This is known as chemotaxis. Okay, there's certain chemicals released from those cell contents that attract neutrophils and macrophages to the area. Chemotaxis is it's like all those cells just make their way towards that area because they're kind of called in by a chemical alarm. Okay, we also get fibrin formation. We just talked about that with blood clotting. This is going to stop bleeding. Okay, and we get pus formation. Pus, that white stuff that we see in our zits and other infected areas, is dead neutrophils, dead bacteria, and debris from the injury. Okay, so little question here: which of the following lead to pain? So this is an interesting one to think through. So cell contents binding to nociceptors, for sure. Talked about that. We can also have pressure that's created by the swelling, pushing on nociceptors. Blood clotting, probably not. Vasodilation, sure, because vasodilation led to the swelling. It also led to immune cells coming, and those immune cells also release things that activate pain receptors. And then chemotaxis, that's all those immune cells coming. They release things that activate the pain receptors, so those for sure. So all those things related to pain, probably not blood clotting, but the rest. All right, so let's walk through this flow chart. I think this can be helpful in you figuring out how this all works. Okay, so we start out with an injury to tissue, right? So we get a splinter into our finger. All right, that's the injury. Okay, so those mast cells interact with things they're not used to interacting with, so they go, uh-oh, what's going on here? We better release histamine. Okay, but then we get other chemicals also from the damaged cells, those cell contents, spill out everything. All right, so what does that lead to? That leads to the three main responses, which are vasodilation. That's directly from histamine, right? Histamine does that. Number two, that leads to increased capillary permeability. That's from the cell contents. And we directly get pain because of cell contents, okay? Um, and we also get chemotaxis, again, because some of those cell contents are stimulating that. Okay, so what does vasodilation lead to? Well, obviously that leads to increased blood flow, and that increased blood flow leads to heat, one of the signs of inflammation, also leads to redness, another sign of inflammation. And we can see that increased blood flow causes delivery of cells and clotting factors, so we get blood clotting. Also leads to clearing out of debris and toxins, which is good. Okay, and then we see this line going over here, increased blood flow is going to lead to swelling as well, right? We get more fluid sent to that area. Secondly, we got, so this would be kind of 1 here. 2 would be increased capillary permeability. This allows fluid and leukocytes to enter the interstitial space. So again, that helps us with delivery of cells and clotting factors. Also helps us to clear things out. Also is partially responsible for the swelling that we see. Okay, third main response is chemotaxis. That's immune cells migrating and coming to the area. Okay, so we get leukocyte and phagocyte migration, which that's going to be part of the swelling we see as well. And so that's a very nice thing for you to look at there and figure that out, work through that till you understand it. Okay, so what leads to swelling? Which ones? Obviously the vasodilation, it's getting more fluid, definitely capillary permeability, not blood clotting, but yes, chemotaxis as well, because that's part of the swelling, is those immune cells. All right, and then lastly, fever. Fever is a systemic inflammation. When we get inflammation through our whole body, that leads to a fever. Our body temperature is normally very strongly maintained at 98.6 by an internal thermostat, somewhat like this, in our hypothalamus. If our body gets too cold, if it goes too much below this, then we increase our metabolism to produce more heat, and we can shiver as well, which produces a lot of heat. If a body gets too hot, that temperature goes up, then we decrease our metabolism, and we can also sweat to shed some of that extra heat. So what causes the fever are chemicals called pyrogens, which can be released by bacteria, other microbes. They can also be released by our immune cells that are interacting with the antigens. And the effect of this is that it increases our set point of temperature in the hypothalamus. So instead of the thermostat being set at 98.6, those pyrogens hit the hypothalamus and say, no, we're actually supposed to be at 101.7, right? So you immediately will feel cold if you're at 98.6. That's why you get kind of cold. That's the chills part, right? And then your body does these things, increases your metabolism, causes shivering, gets that temperature up to 101.7, okay? And this is good in some ways because this reduces iron levels in your blood. This makes it so microbes can't multiply as well, but it also makes you incredibly tired and uncomfortable. Okay, so then you give yourself some Tylenol, all right, or Advil, and that actually blocks those pyrogens, decreases your set point back to 98.6, but you're actually at 101.7. So that's why we can, so then we do the opposite. We're too hot, so now we move to sweating, right? And this is when we break our fever. We sweat, we totally sweat out our whole bed, maybe in the middle of the night. If you've had the flu, you've experienced this. And then your temperature drops back down to 98.6. Okay, so that's what a fever is all about. Fevers can be dangerous if they last too long or the temperature gets too high. It can destroy proteins, can destroy cells. Too low of iron levels for too long can lead to decreased ability to move oxygen around the body. Fevers can be more extreme in kids and can also lead to what's known as a febrile seizure. So yeah, should we treat them? Mild fevers, we're probably better off just leaving them alone because that actually helps us fight the infection. But with kids, it's hard to tell them that when they're so uncomfortable. It's nicer just to let them be comfortable. And then if it starts getting up too high, then we definitely want to treat it. All right, guys. Hope that's all good for you. Let me know if you got questions. Hop on the office hours or shoot me an email. Talk to you soon. Bye-bye.